Alright guys, so what we're going to do now is introduction to food proteins. Before we introduce you to food proteins and its properties, we have to talk about the protein itself. You should be familiar with this kind of products. This is made from fish protein. They call this the original surimi, that is then mixed with flour and form the fish ball like products. Of course, this is not real fish, all of this. It is a mixture of food ingredients. And then we have tofu. Tofu is basically made from soya. So you can imagine soya protein, the milk of soya protein being coagulated. Can be done by using gluconodelta-lactone, which is a type of acidulant, or you can use things like calcium chloride and similar coagulating agents. Coagulate the proteins and then you remove the water from the system and you have yourself tofu. Egg should be familiar to you. This particular area here, the white has so many different kind of protein. Basically minus water in this white area of the egg is basically all protein. Yeah, some protein inside the yolk, but basically when we eat eggs, we are aiming for the proteins surrounding the yolk. If you are familiar with our local dish, this savory dish, every year we consume bahulu. It is made basically from egg white by using functional properties of egg white, which is it forms some kind of gas or aerated products that is then stabilized by the emulsifier. This is emulsifier actually, lecithin inside the egg yolk, and then as cooking goes by, the egg white form gel and it stabilizes the whole bahulu. So we must understand the protein. Alright, so the basics of protein that you can find in any biochemistry books. It is basically polymers of amino acids, we refer to as polypeptides. Yeah, so the polymers are folded in such a way that they have biological activity. So it is very important to understand the structure of proteins, how these polymers are formed, and why the folding is actually very important for the biological activity of a particular protein. Protein, you know, is the most diverse biomolecules in our body. We have various enzymes that catalyze various chemical reactions. We have receptors that help in the controlling of signals in the body. It's like traffic lights. And then we have carrier like hemoglobin that carries oxygen throughout the bloodstream, or muscle and organ tissues. This provides structure and mobility of the body. Okay, so to form protein, there are amino acids that shares a common structure with amine group NH2 and acid groups COOH. And then in the middle there is central carbon atom bonded to hydrogen to a side chain that's normally written as R. The distinct side chain or groups allow for different classification into, you have acidic, you have basic, you have neutral, you have aromatic, I will show you later, and sulfur containing amino acids. So proteins are composed of amino acids joined by the peptide bonds. I will show you later. The peptide bonds connect the acid end of the one amino acid with the amino ends of another. and when two amino acids bonded together, we call them dipeptide. So on and so forth. You can also form oligopeptides, meaning oligo means just a few, so it can be, you know, maybe 5 or 6, maybe 10 amino acids become oligopeptides. More than 10, you start to form polypeptides. Even though polypeptides can be more longer than 10, can be up to 300 or 1000 amino acids. Yeah. So this is the what we mean by having the polypeptide bonds. Yeah, that's the polypeptide bond there. This is one amino acid called, if I'm not mistaken, this is aspartic acids, and there's another amino acid here, if I'm not mistaken, called phenylalanine. So by a process of condensation reaction that results in the elimination of water, you end up having a dipeptide, which is here, with the formation of the dipeptide bond. Classification of amino acids, as mentioned before, they can be neutrals, valine, glycine, alanine, leucine, isoleucine, serine, and threonine, all neutral. The basics are basically the very famous lysine, because of this particular R groups here, that the N there, NH2, is what makes lysine basic. Similar to lysine is arginine, it has NH2 at the end. Similar to that is histidine, also have NH2 somewhere there. So the opposite of basic is the acidics, aspartic acid, having the acidic groups there, and on the R groups, on the R side chains, also glutamic acid. Okay, and then finally we have sulfur containing amino acids, like if you can see group sulfur there, also here and here. So this is cysteine and methionine and cysteine. This is very famous for in egg white. When somebody leave the egg too long outside, the egg can develop rotten phenomenon and yielding the hydrogen sulfide from the breakdown of these amino acids. Very bad odor. And then we have aromatics amino acid like phenylalanine, tyrosine, and tryptophans. Okay, so the important thing is we understand now that the formation of dipeptide is important for making the amino acid chain longer and longer, and then formation of the polypeptides. We know the amino acids can be classified into 5 categories: neutral, basics, acidics, and sulfur containing, and also aromatics. Alright, so next what we need to do is something more complicated, the structure of amino acids. But we stop for now for part 1 of protein as food component. Thank you guys, so happy learning.